Hello everyone and welcome to today's live broadcast, our keynote presentation, Targeting Molecular Chaperones in Cancer, Lessons Learned from HSP90, presented by Dr. Len Neckers, Senior Investigator at the National Cancer Institute Center for Cancer Research. I'm Susie Valdez of LabRoots and I'll be your moderator for today's event. We're delighted to bring you this educational web seminar presented by LabRoots. LabRoots is the leading scientific social networking website and producer of educational virtual events and webinars. Before we begin, I'd like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the green Q&A button located at the lower left of your presentation window and type those questions into the box that appear on the screen. Dr. Neckers will be following up with your questions via email and answering them in order. Also, please notice that you'll be viewing the presentation in the slide window. To enlarge that window, just click on that screen icon located on the lower right. If you have any trouble hearing or seeing the presentation, click on the support button or the Q&A button and let us know you're having a problem. This presentation is educational and thus offers continuing education credits. Please click on the button in the bottom left-hand corner and follow the process to obtain your credits. Without further ado, please join me in welcoming Dr. Len Neckers. I will now turn the presentation over to him. Welcome, Dr. Neckers.
Today I'm going to talk to you about our experience targeting molecular chaperones in cancer and lessons we've learned from HSP90. In normal cells, HSP90 is involved in a proteostasis network that maintains the viability of cells. In cancer cells, this includes maintenance of a number of mutated and/or activated oncoproteins, including some kinases that are cancer-associated, transcription factors, etc. The importance of HSP90 differs, and dependence on HSP90 differs between normal cells and cancer cells, and I want to give you an example of how we can interpret that. So this is an experiment using yeast grown at optimal temperature with wild-type HSP90. And from this graph, you can see that the viability of these yeast are not affected until the expression level of HSP90 is reduced by as much as 90%. So there's a huge excess of HSP90 under optimal growth conditions in these organisms. Now, if we make a temperature-sensitive mutation to HSP90, as shown in the red box, at optimal growth conditions, there's still no effect on growth, comparing wild-type growth to growth of the W585T mutant. But if you now put the yeast under suboptimal temperature, in this case 16 degrees, you can see that the mutant is unable to support growth, while the wild type and two other mutants are completely able to support growth, showing that while this particular mutation functions perfectly at optimal temperature, it's unable to support yeast growth at suboptimal temperature. Another way of looking at this is looking at expression strength of HSP90, comparing that of wild type to yeast mutants. In particular, we're focusing on the yeast mutant W585T that's in the red box. And you can see as you decrease the expression of this mutant, viability of the yeast, as shown by conversion from the yellow color to the blue color, is compromised. At the same time, a similar reduction in expression of wild-type HSP90 has no effect. So, although these yeast are being grown under optimal conditions, reduced expression of the mutant has consequences for viability. In other words, the yeast become more dependent on high levels of expression of this mutant. So diagrammatically, if you now look at normal cells, they can tolerate a large reduction in the levels of HSP90 without any obvious effects on fitness. Whereas if you stress these cells, they become much more dependent on high expression of HSP90. And if you exchange stress for a cancer, our working hypothesis over the years has been that cancer cells have a greater dependence on HSP90 relative to normal cells, so that a significant portion of this chaperone can be pharmacologically inhibited in normal cells without any obvious effect. But similar inhibition in cancer cells is likely to be cytotoxic. In fact, HSP90 is known to modulate a cell's response to many different kinds of noxious environments that are frequently characteristic of the cancer environment and lifestyle. These include nutrient stress, proteotoxic stress, hypoxia, and genetic instability, frequent characteristics of many cancers. HSP90 clients, those proteins that are chaperoned by HSP90, are integral in allowing cells to respond to these insults. A number of studies have shown, and one is provided here, that amplification and high-level expression of HSP90 is correlated with aggressive phenotypes of different kinds of cancers, including lung cancer and, in this case, HER2-negative breast cancer. So what these data showed in this paper published in 2012 is that looking at survival as a function of HSP90 expression in patients with triple-negative breast cancer shows that patients whose tumors have upregulated HSP90 expression show significantly poorer overall survival compared to patients whose tumors have less HSP90 expression. And again, this has been repeated over the years for many kinds of cancers. So what does inhibiting HSP90 do? From what I've shown you so far, and based on yeast models, the likelihood is that, or the expectation is that inhibiting HSP90 compromises the robustness of a cancer cell. And some preliminary evidence that this is the case was shown in a paper that we published now a number of years ago using HSP90 inhibitors, in this case the first HSP90 inhibitor in clinical trial, 17-AAG, as a radiosensitizing agent. So there are 3 cervical cancer cell lines here, and what we're showing is that in each case, inclusion of a small amount of HSP90 inhibitor is able to promote radiosensitization in vitro of these cells. And this radiosensitization is in fact dose-dependent and time-dependent. So how does HSP90 function? Understanding this is critical to understanding how pharmacologic HSP90 inhibitors work. ATP binding and ATP hydrolysis are essential for the function of HSP90 in vitro and in vivo. Looking at the crystal structure of HSP90, this is just focusing in on the end domain of the protein. We can see there's an ATP binding pocket And this ATP binding pocket turns out to be the binding site of two of the original natural product HSP90 inhibitors that were identified, galdanamycin and radicicol. So when HSP90, which is a dimer, exists in the absence of nucleotide, it's considered to be in what's called an open conformation, where the end domains are undimerized, as shown here. In the presence of ATP, the end domains close, a number of conformational changes occur, and ATP binding and hydrolysis is important for driving this conformational cycle. In the presence of HSP90 inhibitors, ATP can't bind, HSP90 remains in an open conformation, and the consequences of this for client proteins is that they're ubiquitinated by E3 proteins associating with these chaperones, and ultimately degraded by the proteasome. We've identified two such E3 ubiquitin ligases that associate with both HSP90 and HSP70. One is CHIP, and the other is Cullen-5. Development of HSP90 inhibitors has gone on since the early 90s, and we're showing here the increased number of HSP90 inhibitors, all binding to the ATP pocket, that have entered clinical trial since the late 1990s. Unfortunately, inhibitors that are currently in clinical trial are only represented by a few candidates, as shown on this slide, including SNX 5422, AT13387, Exelixis 888, PUH71, TAS116, and a new compound, which I'll talk about towards the end of the talk, PEN866. No HSP90 inhibitor has yet been approved by the FDA, although active trials of HSP90 inhibitors of these agents currently in clinical trial is shown here, and you can see that many of these studies involve combining HSP90 inhibitors with other agents. Some anecdotal studies clearly show that HSP90 inhibitors are effective clinically in certain kinds of cancers. This is a relatively early study using 17-AAG in HER2-positive breast cancer patients who stopped responding to the antibody Herceptin. When 17-AAG was included along with Herceptin, there was a significant response rate of 26% and an overall clinical benefit rate of 63% in these patients whose cancers were otherwise no longer responding to treatment. Similarly, in a phase 2 study of non-small cell lung cancer, another HSP90 inhibitor used as monotherapy showed significant responses in ALK-positive non-small cell lung cancer and in KRAS mutant small cell lung cancer. This is an example of a patient's tumor shown at baseline on the left-hand side in the red circles and arrows, and after 3 weeks of gantenerumab treatment, and gantenerumab is the HSP90 inhibitor used in this case. And it should be noted that this patient previously progressed on chemotherapy, received the ALK inhibitor crizotinib for 1 year, and then progressed on that, and then and only then enrolled in this gantenerumab monotherapy trial and showed clear responses. Gantenerumab activity has also been seen in mutant BRAF non-small cell lung cancer, again after the patient progressed on various forms of chemotherapy. Single agent response to HSP90 inhibitor is clearly evident in the images below. Lastly, this slide represents a targeted approach beyond ALK, and again, this is triple-negative breast cancer, which I previously showed you a few slides ago, is aggressively related to high expression of HSP90. And here again, single-agent treatment of a patient with gantenerumab who had stopped responding and progressed on various forms of chemotherapy show clear responses. So these data would suggest that HSP90 inhibition is in fact able to compromise the fitness of cancer cells and should show reproducible and robust clinical activity. However, since no drug showed sufficient activity so far to be approved by the FDA, we and others have looked into this question further and have noticed that another consequence of HSP90 inhibition is to induce what is known as the heat shock response by potentiating activity of the master transcriptional regulator of the heat shock response, heat shock factor 1, or HSF1. The heat shock response, in a way, is poorly named because it's initiated by many types of proteotoxic stress, including elevated temperature, oxidative stress, exposure to heavy metals, and bacterial or viral infection. But upon activation of the heat shock response, which is mediated by HSF1, one sees an upregulation of proteins involved in proteostasis, including the heat shock proteins HSP70, HSP27, and HSP90 itself. And if HSP90 inhibitors activate or potentiate HSF1, this becomes a potential problem in cancer treatment. This is a Western blot, which I want to show you as an example in ERBB2 overexpressing breast cancer cells. So here we're using the prototypical HSP90 inhibitor galdanamycin, and we're using a dose response to look at loss of ERBB2 protein due to degradation in the proteasome, and looking to see when we see induction of HSP70. And as you can see, as soon as there's an effect on the client ERBB2, there's an upregulation of HSP70 protein expression. Another HSP90 dependent client, phospho or activated AKT, is similarly affected at an equivalent dose that induces HSP70. Importantly, as shown in the bottom two panels, HSC70, the constitutive isoform of HSP70, is unaffected by HSP90 inhibitor treatment. So only the stress-inducible form, HSP70, is affected, and this is consistent with dependence on HSF1 expression. In fact, looking at levels of HSP70, mRNA or protein, has been used as a pharmacodynamic marker in patients on HSP90 inhibitor clinical trials. So you can see from this figure that there is a dose and time-dependent increase in HSP70 in patient blood after treatment. And it depends on how many cycles of treatment. The more cycles of treatment, the higher the induction of HSP70 is. And this has actually been used as a biomarker in clinical trials to show that we're hitting the target. But in retrospect, this appears to be an indication that we may be obviating the effect of the anti-cancer activity of HSP90 inhibitors by promoting this tissue protective response. To further complicate matters, the Lindquist lab published a study a number of years ago showing that in addition to inducing a cytoprotective heat shock response in all cells, including cancer as well as normal, HSF1 drives a cancer-specific set of genes that promote the growth and viability of cancer cells. So this is an additional reason why one would not want to upregulate or activate HSF1. In fact, if one looks at HSP70 expression in tumor, one sees that, like HSP90, it correlates with poor survival. So this is a study of survival in breast cancer where the HSF1 status of the patient's tumor has been evaluated. And there's a highly statistically significant correlative effect of HSF1 expression shown by the red line and poorer survival compared to low or no HSF1 expression in tumors shown by the black line. Agreeing with this observation, if one looks at TCGA data that has recently appeared and is available, one sees that HSF1 is overexpressed significantly in a wide variety of cancers. Comparing its overexpression to MYC, which is frequently overexpressed in cancers, you can see that in many of the various cancers shown here, and they're delineated on the x-axis, HSF1 overexpression is greater and more pronounced across this range of cancers than is c-MYC. So it's becoming very apparent that cancer cells depend on HSF1 for growth, viability, and survival, and that a targeted agent that results in increasing the activity of HSF1 is not going to promote an anti-cancer response. We wondered what the mechanism behind HSP90 effects on HSF1 is, because understanding the mechanism may allow us to intercede and block the effects of HSP90. So the current model that's been in the literature for many years is that HSP90 binds to monomers of HSF1 in unstressed cells, and in the presence of misfolded proteins caused by proteotoxic stress, such as heat shock, HSP90 comes off of HSF1 to deal with the misfolded proteins and allows HSF1 to trimerize, which it must do to become an active transcription factor. So how do HSP90 inhibitors affect the HSR, or heat shock response, based on this model? The concept was that HSP90 inhibitors, by preventing ATP from binding to HSP90, cause dissociation of HSP90 from monomeric HSF1, and similarly to heat shock, allow HSF1 to trimerize and become an active transcription factor. So the dogma in the literature is that HSP90 functions as a repressor of HSF1 activation. It functions as a sensor of proteotoxic stress, and release of HSF1 from HSP90 is a key event, if not the key event, in induction of the heat shock response. So we asked ourselves, does this model make sense? And in fact, we came to the conclusion that it does not. First of all, it's extremely difficult to visualize the interaction of endogenous HSP90 and HSF1 in cells without chemical cross-linking. And a recent study, in fact, in vitro, showed that the constitutive isoform of HSP90 actually promotes temperature-dependent trimerization of HSF1, inconsistent with the current model. So we decided to examine more closely if we could detect a stable interaction of HSP90 with HSF1. And to do that, we used two sets of mutants. The first set of mutants keeps HSP90 in an open conformation by preventing ATP binding. The second set of mutants, shown on the right, keeps HSP90 in a closed conformation by allowing ATP binding but preventing ATP hydrolysis. We've made both sets of mutations in HSP90 alpha, the stress-inducible form of HSP90, and beta, the constitutively expressed form of HSP90. And what we found, as many others have found, is that wild-type HSP90 is not able to be detected associating with HSF1 without cross-linking. However, the closed mutant D47A of HSP90 alpha readily associates with HSF1 in immunoprecipitation assays. The closed mutant of the beta isoform also associates, but not as strongly as stress-inducible HSP90 alpha. So what this is telling us is that the ATP-bound HSP90 can associate, and its association is readily detectable with HSP90. But in human HSP90, the ATP-dependent closed conformation is very transient and is only infrequently accessed by the chaperone, even in the presence of ATP. We next look to see whether the impact of HSP90 inhibitors on this interaction was similar to other inducers of the heat shock response, including heat shock itself as well as proteasome inhibition. So what we found is 3 different HSP90 inhibitors that have been in clinical trial all dissociate HSF1 from this closed HSP90 alpha mutant, as shown on the left. However, neither the proteasome inhibitor MG132 nor heat shock itself dissociates the interaction of closed HSP90 with HSF1, making the point that HSF1 dissociation from HSP90 is clearly not a prerequisite for induction of the HSR. So what HSP90 inhibitors are doing is unique. So how can we explain what HSP90 inhibitors are doing to potentiate the heat shock response? A clue came from this experiment where we're looking at HSP70 reporter activity in cells treated either with 17-AG alone, heat shock alone, or the combination. And then they're allowed to recover for 4 hours at normal temperature. And you can see that 17-AG alone has a minimal effect on HSP70-driven reporter activity. Heat shock has an effect, as expected, but the combination of 17-AG and heat shock has a synergistic effect that is much higher than one would expect from just an additive response. So this led us to ask the question, perhaps HSP90, and particularly HSP90 alpha, may be working to shut off the heat shock response as a feedback mechanism, and that by dissociating HSP90 alpha from HSF1 bound to HSF1 elements in promoters, this results in potentiation of transcriptional activity and prolongation of the heat shock response. To examine this hypothesis, we did an HSF1 chromatin immunoprecipitation assay, shown here, and we looked at HSF1 binding to both the HSP70 promoter as well as the HSP90 promoter, two genes that are regulated by HSF1. And we either exposed cells to heat shock alone for 30 minutes or first pre-treated with the HSP90 inhibitor 17-AAG for 2 hours, followed by 30 minutes of heat shock, and then allowed recovery from anywhere from 5 minutes to 6 hours, and monitored by chip association of HSF1 with DNA. So what you can see in the absence of 17-AAG, if you look at both the HSP70 promoter shown in blue and the HSP90 promoter in red, that N, of course, represents no heat shock, and that's set to 1. So all these values are in relation to no heat shock. After 5 minutes of recovery from heat shock, you see a big increase in HSF1 bound to both HSP70 in particular, but also HSP90 promoter regions. But this effect is lost by 2 hours, where HSF binding goes below baseline and remains below baseline for the duration of the experiment. However, if cells are pretreated with 17-AAG prior to heat shock, you see an increase in HSF1 binding in the absence of heat shock and a continuation of this binding during the recovery for a much longer time than in cells treated with heat shock alone. So binding above control persists for at least 4 hours, if not longer. And this results in increased HSF1 dependent transcription as well as translation, and can explain the induction of HSP70 caused by HSP90 inhibitors that I showed you before. Based on these data, we've come up with a working model to explain what HSP90 is doing to regulate the heat shock response. We think that HSP90 alpha in particular, as an HSF1 target gene, serves to provide feedback control to the heat shock response by binding to HSF1 and removing it from DNA. HSP90 inhibitors, in contrast, by dissociating HSP90 alpha from HSF1, prevent this from happening, block the feedback control, and extend the duration of HSF1 transcriptional activity bound to DNA. Understanding this mechanism as perhaps the true model of what HSP90 inhibitors are doing is allowing us to look for novel strategies to disrupt the ability of HSP90 inhibitors to dissociate HSP90 from HSF1 or to otherwise block HSF1 transcriptional activity. For the last part of this talk, I want to describe a novel use of HSP90 inhibitors that does not involve its ability to degrade client proteins, but only to deliver cytotoxic drugs to tumor cells and to prolong their duration within the tumor cell. So one of the characteristics of HSP90 inhibitors as a class has been known for many years to localize to tumor cells and to remain at relatively high concentration in tumor cells for much longer than in normal tissues. So researchers at Synta Pharmaceuticals designed a a conjugate approach where they attached a linker to their HSP90 inhibitor and coupled their HSP90 inhibitor to the active metabolite of irinotecan. Irinotecan is a topoisomerase 1 inhibitor whose active metabolite SN38 is highly cytotoxic and causes significant toxicity to normal tissues. So the folks at Cinta hypothesized that by delivering this toxic molecule directly to tumor cells and maintaining it at relatively high concentrations in tumor cells for an extended time because of its attachment to HSP90 might not only improve its efficacy toward tumor inhibition or killing, but also reduce its systemic toxicity. And this has in fact proved to be the case. In collaboration with Lee Hellman and Christine Heskey in the Pediatric Oncology branch here at NCI, we've looked at use of this conjugate in animal models of Ewing sarcoma and rhabdomyosarcoma, two pediatric sarcomas that are frequently treated with irinotecan, but invariably stop responding after a certain time. Shown in these graphs are the responses of a rhabdomyosarcoma line, RH30, to either HSP90 inhibitor alone, which is gantenerumab in this case, shown in the red line in panel A, to irinotecan alone, shown in the purple line, and to 3 different doses of this conjugate that I've discussed, shown in the yellow, blue, and dark purple lines. As you can see, HSP90 inhibitor itself is ineffective in inhibiting the growth of RH30 xenografts in mice, while irinotecan, although initially very effective, eventually loses activity and the tumors return. In contrast, the drug conjugate, in a dose-dependent manner, is able to significantly prolong regression of these tumors. The result is even more dramatic in a Ewing sarcoma model shown in panel B, where you can see again that gantenerumab alone, the HSP90 inhibitor, is ineffective. Irinotecan, as expected, is very effective, but its anti-cancer activity is transient. And again, there's a dose-dependent effect of the conjugate such that at doses that are achievable in humans, there's essentially no recurrence of this cancer after 2 treatments. This is also observed in a Ewing sarcoma model subjected to protracted therapy with irinotecan compared to the conjugate. As you can see, even though irinotecan treatment is protracted, tumors eventually will regrow, but 2 treatments with the conjugated HSP90 irinotecan active metabolite cause complete regression in this tumor model and complete survival of the mice. Lastly, this is a Ewing sarcoma PDX model, otherwise naive to irinotecan. You can see that the irinotecan response shown in purple lasts for a significant amount of time, but eventually tumors become non-responsive. Addition of HSP90 inhibitor together with the irinotecan, but as 2 separate agents, has no significant benefit. But again, the conjugate of HSP90 inhibitor coupled to irinotecan shows much better activity. We looked in these tumors at markers of irinotecan activity, one of which is gamma H2AX, an indicator of DNA damage caused by irinotecan. And in panels A and B, you can see that expression of gamma H2AX, an indicator of ongoing DNA damage, persists far longer in the conjugate-treated animals than in animals treated with irinotecan alone. And lastly, in the bottom panel, we looked at a series of genes that are either upregulated or downregulated in response to irinotecan. And the irinotecan alone samples are shown in orange, and the conjugate samples are shown in blue. And you can see that in both cases, but actually most obviously in those genes that are downregulated in response to irinotecan, the conjugate is significantly more active. These exciting data have led to initiation of a first-in-human clinical trial of this conjugate termed PEN 866, sponsored by Tarveda Therapeutics, opening at the NCI and other sites in July of this year. And this is now open for enrollment. With that, I'll stop, and I'd like to acknowledge the folks in my lab that participated in these experiments, other investigators in NCI whose collaboration was essential to our work, and collaborators from Synta Pharmaceuticals who initially supplied us with the conjugate of HSP90 inhibitor and irinotecan. Thank you very much for listening.